In this video lecture, I will be talking on T-cell receptor diversity. And as you may know that T-cell receptor should be able to recognize around 10 to power 16 of different microbes or pathogens. But however, T-cell receptors DNA has fewer variable gene segments, around approximately 3,640 variable gene pairs. But however, there are several possible mechanisms contribute and generate high degree of T-cell receptor diversity. For example, random gene segments rearrangement, junctional diversity in form of B and N nucleotide addition. But however, I find it would be interesting to this lecture to give a short account on allelic exclusion, which ensure expression of one allele of gene. And as can be seen here, delta gene segments, they are located within the alpha gene complex. So for a successful alpha gene rearrangement, delta gene segments should be deleted by alpha chain rearrangement. That is to say, exclusion of delta genes located on the same chromosome. But however, for B cell, and as can be seen here, this is antibody, and as you know that antibody, it has two symmetrical heavy chains and two symmetrical light chains. For the specificity and to ensure single specificity, heavy chains should come from one allele, either from maternal or paternal alleles, and this is true for the light chain, and this is to ensure a single specificity. It is very important that rearrangement take place in the right direction. The rearrangement is directed by recombination signal sequence, which are located 3 sides of each V or variable gene segment. This is a variable gene segment, and this is the 3 side. As can be seen here, it is 23 base pair, and also it is located at the 5 side of the joining segment. This is beta, actually gene segments or gene segments that encode the beta chain. But however, they flank the diversity, I mean the recombination signal sequences. As can be seen here, the D segment is flanked by recombination signal sequences from both sites, and these sequences redirect the rearrangement according to the role of 12 to 23. So now we can see this role. The role of 12 base pair to 23 base pair. Let us first see or look at the recombination signal sequence. As it can be seen here, there is a spacer, which it could be 12 base pair or 23 base pair. So random gene segments recombination is directed by the spacer of the recombination signal sequence, which could be 12 base pair or 23 base pair. And as can be seen here, this is TCR alpha gene segments flanked by recombination signal sequence spacer of 12 base pair. It flanks the joining segment, and 23 base pair, it flanks the variable alpha, segment. For TCR beta gene segments, as can be seen here, the recombination signal sequence of 23 base pair spacer flank the variable beta at the 3 side and flanks the joining beta segment at the 5 side. But however, the D beta segment is flanked by 12 base pair spacer or 12 base pair recombination signal sequence at both sides. And according to the role of 12 base pair to 23 base pair, the rearrangement of TCR alpha gene segments, it will take the direction from the joining alpha, as it is flanked by 12 base pair, towards the variable alpha, as it is flanked by 23 base pair recombination signal sequence. In case of TCR beta gene segments, the direction of gene rearrangement, it takes place, as can be seen here, the D or diverse segment, it is flanked by 12 base pair on both sides, so the direction will be from 12 to 23, and this will lead to formation of DJ, and then DJ will follow this direction to the 23 base pair that flanks the variable beta, as it is flanked by the 12 base pair. So now I am going to talk about the how the TCR gene rearrangement is initiated. Actually, TCR gene segments recombination or rearrangement is initiated by rearrangement activity gene 1 and rearrangement activity gene 2, or RAG1 and RAG2. And as can be seen here, RAG1 and 2, they bind recombination signal sequence. This is for the gene segments for the alpha chain. This is alpha chain. So it binds here, this is RAG, it binds 12 base pair recombination signal sequence, and also it binds 23 base pair recombination signal sequence. So according to the role of 12 base pair to 23 base pair, the G will bind the variable segment or variable alpha, as can be seen here. Then the rag will sit at this place in between actually these two segments, and these two segments will sit like this together. Then rag start to cut or to cleave at these places, and this will end with formation of hairpin due to B nucleotide addition by rag enzymes. However, rag enzymes cleaves the hairpin, and it will end with an injury or incomplete DNA like this. But another enzyme, which is terminal deoxynucleotide or nucleotide transferase, together with Artemis, will add N nucleotides just to repair this injury, as gene should not be left open, and this will lead to formation of a functional gene that has the capacity to encode TCR alpha of the T cell. In gene segments encode beta TCR, RAG1 and RAG2 bind recombination signal sequence 23 base pair, as can be seen here, that flank the three sides of the variable gene segment and 5 sides of the joining beta gene segment. And recombination signal sequence of 12 base pair spacer, as can be seen here, flanks each side of diversity or D gene segment. That is to say, a gene segment flanked by 12 base pair recombination signal sequence can bind only gene segment flanked by 23 base pair recombination signal sequence according to the role of 12 base pair to 23 base pair. And according to this role, now this segment, they bind, this is 12 flanked by the 12 base pair recombination signal sequence. It binds the joining beta, and this will lead to formation of D beta, G beta, or G beta. Then DG, according to this role, will in turn bind to this I mean DG will bind to variable beta as it is flanked by 23 base pair recombination signal sequence. So this will lead to formation of a functional gene that has the capacity to encode TCR beta of the T cell. And now I'm going to talk about junctional diversity. And as you may know that junctional diversity is beneficial for creating diversity array of different TCR required for recognition of around 10 to power 16 of different micro or different organisms. Let us go up again. 10 to power, they should recognize 10 to power 16 of different pathogens. And as you may know that T lymphocyte is a blind cell. So T lymphocytes do not recognize or do not see or cannot see tertiary protein, for example, bacteria. So tertiary proteins, bacteria, will be seen by the antigen presenting cells, dendritic cell, B cell, and macrophage. So they will be taken by this antigen presenting cell. Then they get processed inside these antigen presenting cells and get expressed on the surface in association with major histocompatibility class 2. Actually, this processed protein is a primary protein. Now T cell can see the primary protein. So junctional diversity is very important for the TCR to be able to recognize this large amount of the microbes of different or this large amount of microbes or pathogens. For that reason, junctional diversity is very important for the TCR or for creation of the TCR diversity. So there are several possible mechanisms contribute to generation of high degree of TCR diversity. For example, only junctional diversity, and as I mentioned that it is due to B and N nucleotide addition, and this B and N, the palindromic nucleotide addition, it takes place here when this get cleaved by the RAG1 or by the RAG enzymes, which lead to formation of hairpins. And now these enzymes, I mean RAG enzymes, they will add palindromic nucleotide in this, which led to formation of hairpin. But however, RAG's enzyme cleave this hairpin, and this will lead to, as can be seen here, just to make it clear for you, that for example this is a gene segment and this is another gene segment. So at the end of the coding segment, the RAG will add palindromic nucleotides like this, and at this end it will add also a palindromic nucleotide. This will leave the gene incomplete. So to be completed, other enzymes, for example terminal deoxynucleotidyl transferase, will add randomly N nucleotides to repair the gene, but however in a non-template. These are non-template, and these are templated. For example, TCGA, it will give you on the opposite side TCGA, the same number and the same nucleotides, but here they are a bit different, and it is different from one TCR to another TCR, as can be seen here. So let us now go back to the junctional diversity here and why it is very important. So according to BN nucleotide addition, junctional diversity increases the recognition capacity of TCR to around 10 to power 13. 10 to power 13. Because the aim of this junctional diversity is just to increase the TCR diversity up to 10 to power 16. So junctional diversity alone, actually, it will increase the diversity to around 10 to power 13. That means TCR, they can see up to 10 to power 13 different pathogens or processed antigens. And that is to say, junctional diversity will provide 10 to power 13 plus random gene segment rearrangement, that it will actually provide 10 to power 3. As I mentioned, around there are around 3,640 variable pairs, and this will lead to increase of the diversity up to 10 to power 16. So TCR will be able to recognize different pathogens up to 10 to power 16 or 10 up to 10 to power 16 processed antigens by the mechanism of junctional flexibility. So junctional flexibility is created as a junction of the V and J. This is for alpha gene segment, this is joining alpha and variable alpha. So this the junctional diversity is created at the junction of variable and joining segments where nucleotides, for example B and N, are added. So junctional diversity occur. This is the junctional diversity occurs for alpha gene segments. BN nucleotides will be added in the junction between VDJ, and this is for the beta gene segments of the rearranged TCR beta gene, and between variable and joining gene segments in rearranged TCR alpha genes. RAG1 and RAG2 together with DNA repair protein, for example Artemis, cleave hairpin loops and add B nucleotides. Then terminal deoxynucleotidyl transferase add N nucleotide. And this is actually very clear here, and as can be seen here, this is the gene and these are the coding end where RAG enzymes cleave and then add palindromic nucleotides, and then the deficit in the gene will be buffed by random addition of N nucleotides by terminal deoxynucleotidyl transferase. So B nucleotides derived from coding end of gene segments, as I mentioned. And terminal deoxynucleotide transferase adds N nucleotide randomly to the B nucleotide end of a single stranded DNA before annealing. In summary, the RAG enzymes cut the DNA and form hairpin at the end of diversity segment and joining segment, and RAG enzymes going to cleave hairpin randomly, as I mentioned here. They cleave hairpin randomly. Then terminal deoxynucleotide transferase will add nucleotides known as N nucleotides randomly between gene segments, as can be seen here. B nucleotides will be generated from the hairpin. That is to say, the joining between two segments is generated in a random way, and this is known as junctional diversity, which increase the T cell receptor diversity up to approximately 10 to power 13. Thank you very much, Professor